All right, everyone, welcome and good evening. We are right at the top of the hour, so we're going to go ahead and get started with the webinar. This evening, I am excited to bring you the first session in the Immune Deficiency Foundation's Decoding PI series. Tonight's featured topic is Arming Yourself Against COVID, Flu, and RSV, led by Dr. Michael Keller and Dr. Brant Ward from Children's National. My name is Emma Mertens, and I'm the Program Manager for Education at the Immune Deficiency Foundation. On behalf of IDF, we thank you for tuning in to this virtual event. Through this series, guest presenters will provide a deep dive into clinically advanced topics from the world of PI. If time permits, the session will be followed by an audience Q&A with our presenters. We hope through your participation in these programs that we may move forward in our mission to engage and empower the PI community through education. Here are a few items to keep in mind for the webinar. This evening, we are using the Zoom webinar feature, and attendees should be able to see the slides and hear the presenter and host speak. If you wish to enable closed captioning, click Show Captions on your Zoom toolbar. You can view the full transcript by clicking on Show Captions and navigating to View Full Transcript. From there, the transcript will appear in real time on the right side of your screen. Attendees will not be able to activate their video camera or their microphone. That means all your questions will need to be typed in the Q&A box in the control panel of your screen. They will be read aloud on your behalf. Please refer to the ground rules for Q&A shared in the chat box. A recording of tonight's webinar will also be published on our YouTube channel. We appreciate your patience as it will take about 2 weeks for that upload to be completed. And to visit our YouTube channel, you can see the link being shared in the chat right now. A brief disclaimer. Please remember the information shared during this meeting is not intended to be a substitute for medical advice, diagnosis, or treatment. We are here today as a trusted source and friend to provide you with information. Always seek the advice of your physician or other qualified health provider with questions concerning a medical condition. Never disregard professional medical advice or delay seeking it based on information presented during an educational event. This webinar is made possible by our wonderful sponsors. It is due to their partnership and contributions that we can provide programs like this for the PI community. Please join me in thanking today's sponsors. And now I am so pleased to introduce our presenters for this evening, who are both joining us from Children's National in Washington, D.C. Dr. Michael Keller is a pediatric immunologist specializing in rare primary and secondary immune disorders in children. He is actively involved in research to advance treatments for complex pediatric conditions, focusing on the use of adaptive T-cell therapies for treating both PI and cancer in children. Dr. Brant Ward is the Clinical Director of Immunology at Children's National, specializing in PI, autoimmune, and allergic conditions in pediatric patients. His work concentrates on understanding the underlying causes of immune dysfunction in children and developing innovative therapeutic approaches to treat these conditions. Welcome, Dr. Keller and Dr. Ward. All right, I'm going to stop sharing my slides, Dr. Keller, and then I will pull yours up. Then we'll go ahead and get started.
Sounds great. Huge thanks to the IDF for this invitation. We are very honored to be here, and hope everyone's doing well.
All right, give me just a moment. All right, Dr. Keller, take it away.
Very good. So I'm going to start first presentation by talking about vaccinations in primary immunodeficiency disorders. So as Emma mentioned, I'm a pediatrician and immunologist at Children's National, and Yeah, we can go. And here's my disclosures. So I am a author on the UpToDate website, so I get royalties from them. I'm also funded by both NIH and Jeffrey Nobel Foundation, but I have no conflicts related to vaccines. Don't serve on any boards for industry. So to start off, next to sanitation, vaccines are been identified as one of the most beneficial medical inventions in human history. And the elimination of poliomyelitis from the US and the worldwide eradication of smallpox remain among the greatest medical success stories in history. And during this, during the past century, mortality rates due to infections dropped from a leading cause of death at the turn of the 20th century to falling outside of the top 10. Widespread vaccination and improved antibiotics have turned deadly childhood diseases into largely historical footnotes. Unfortunately, this trend in decreasing mortality due to infectious diseases was reversed transiently during the COVID-19 pandemic. I mean, you could probably zip, I think, the next, but luckily that has reversed in recent years, again, largely due to the rapid rollout of active vaccinations. So I'm going to talk a little bit to start about how vaccines work. So the primary goal of immunization is to produce immune memory. This depends on functioning immune cells, such as T cells. We can scroll forward, which destroy virus-infected cells, and B cells, which produce immunoglobulin proteins to help clear viruses and other infections. And the T cells and B cells work together in order to generate immune memory. And vaccines are really divided broadly into non-live vaccines, which can be either inactivated or recombinant. These include things like polio, inactivated poliovirus, hepatitis, there are diphtheria, tetanus, pertussis, our pneumococcal vaccines, flu vaccine, and now COVID-19. And then there's live attenuated vaccines. Here we can zip forward. Sorry, include things like rotavirus vaccine or measles, mumps, rubella, and the Varivax vaccine. For travelers, this also includes yellow fever vaccine and the oral typhoid vaccine. We can go for it. There we go. So vaccines work on a population level by interrupting transmission of infections. So if a new pathogen enters a population where no one is immune, and here I'm showing you this figure, blue is non-immune but still healthy, yellow is immunized and healthy, and red is non-immunized and sick and contagious. So if you have a largely blue population and there's a new infection, it spreads very widely. If, on the other hand, some individuals are protected by vaccination, they're protected, as shown in yellow, but transmission to others is not impacted. Once the majority of the population is protected, though, then transmission can be interrupted. So the percentage of the population that needs to be immune to prevent transmission depends on a lot of factors, including how infectious the disease is, whether it can establish chronic infections, and for example, for influenza, roughly 2 people are infected for every sick individual. Measles comparatively can spread to as many as 20 susceptible individuals for each sick person, and therefore requires a much higher level of herd immunity to prevent transmission. So by maintaining this level of population immunity, we can secondarily protect immunocompromised patients as well as the very young and very old who can't receive these vaccines and are susceptible. So, importantly, with regard to susceptible populations, roughly 500,000 children in the U.S. are too young to receive all vaccines due to their age, and particularly is the case for live vaccines such as measles, mumps, and rubella. And for that population, herd immunity is very critical. And for many years, there was very few measles cases, usually almost exclusively travel associated in the U.S. But unfortunately, there have been periods of resurgence that many of you probably remember, including transmission that occurred in Florida in 2019. Obviously, that dropped off during the pandemic, but we're still seeing a fair number of cases annually, and that largely in 96% of the cases that occurred last year were in patients who were either incompletely vaccinated or not, or their vaccine status is unknown. Vaccinations in the setting of immunology is also important to better help us to understand how the immune system works. So by looking at responses to vaccines, we can help us to determine how well our immune memory and through that our T cells and B cells actually function. And obviously, many types of immune deficiencies impair our ability to respond to vaccines. And importantly, though the vast majority of inactivated vaccines are quite safe, certain vaccines, particularly live vaccines, can actually be harmful to certain special populations. And that includes patients with certain types of more severe primary immunodeficiency, patients undergoing bone marrow or solid organ transplantation, patients with very advanced HIV, or patients who are receiving strong immunosuppressive medication for rheumatologic or inflammatory diseases. So, in primary immunodeficiency disorders, disease due to live vaccines have been described, albeit rarely, in patients who receive live vaccines. This can include, we can scroll forward, uh, the BCG vaccine, which is given internationally but not in the US to prevent tuberculosis, um, as well as the rotavirus vaccine, and in rare cases, the measles, mumps, rubella vaccine. I'll talk a little bit more about the specific cases here, but essentially all of these are occur almost exclusively in fairly in moderate to severe types of immune deficiency. Usually those that impair the T cells. So in 2014, an Immunodeficiency Foundation expert panel provided vaccine recommendations for patients with primary immunodeficiency based on all of available evidence. And I've noted a new vaccine guideline is currently being developed by the American Academy of Asthma, Allergy, and Immunology. And that is in the works and hopefully will be out by 2025. But we're going to go over the details of this with a couple of specific updates related to new vaccines that are available. So we'll go over these bispecific conditions. So starting with the more severe types of PID, specifically the severe T cell defects like severe combined immune deficiency and related diseases like athymia due to complete DiGeorge syndrome. So in these patients, live vaccines would be strictly contraindicated. So you can click forward. So we would not give these patients live vaccines because we know in some cases they can be harmful. Inactivated vaccines are likely safe, but usually are ineffective because if you don't have immunity, you won't have good B cell function. And most patients with these disorders would be receiving immunoglobulin and thereby getting passive immunity to many of these pathogens anyway. No. In patients with mild to moderate T cell defects, like for instance, the non-incomplete DiGeorge syndrome, dactyl-telangiectasia, cardiovascular hypoplasia, or other forms of combined immunodeficiency. Selective live vaccines may be safe, but it depends greatly on individual cases. So really having evaluation with an immunologist is really critical before a person could be cleared for these. Inactivated vaccines are safe and may be beneficial depending on someone's function. And again, since even within these disorders, there's a spectrum of disease, it's important to really know to really understand details of someone's immune function. Importantly, related to this, there have been descriptions of deep tissue and skin inflammatory lesions called granulomas in patients with different forms of T cell deficiency, which occurred anywhere from early childhood up until early adulthood. And several studies have established that in these granulomas in patients with T cell deficiencies, that rubella virus that was tied back to their original MMR vaccination was found in some of these lesions. And some of these cases, these inflammatory complications have been very difficult to control, and in some cases, even been an indication for patients to undergo a bone marrow transplant to replace their immune system, which has been effective in controlling this. Uh, so moving forward to patients with B-cell deficiency, and this includes patients with agammaglobulinemia, common variable immune deficiency, or related antibody deficiencies. Most live vaccines are generally considered contraindicated in these cases, and again, most of them wouldn't be effective. Inactivated vaccines are likely safe. In most cases, may not be wholly effective. Um, and in patients who are getting immunoglobulin, they should be getting passive protection against these. There are a couple of exceptions, though, that we'll talk about, including influenza and COVID-19. You can click forward. All right. So in, in patients who are receiving immunoglobulin, um, there is a protection against many vaccine preventable illnesses in which would be detectable in the plasma donors who donate to make these immunoglobulin products. Really, the only exceptions are for pathogens that are more rapidly shifting, like influenza vaccine, now COVID-19, as well as pathogens that are mostly controlled by T cell immunity. So generally speaking, and we'll talk about the evidence in a moment, we do recommend the inactivated influenza vaccine as well as COVID-19 boosters in our patients with antibody deficiency, even if they're getting immunoglobulin. We also will generally recommend other vaccines that are inactivated that may engender a T cell response, as that may still be valuable in patients with B cell deficiency. So um, previous studies have looked at both antibody and T cell responses to vaccinations in patients with antibody deficiency, including CVID. So a study out of Europe looked at responses to an activated typhoid vaccine and showed that 41% of patients did respond to it. So in terms of influenza vaccines, several studies internationally have also looked at both antibody and T cell responses to flu vaccines and showed that roughly 20 to 30% of patients had detectable responses to an inactivated influenza vaccine. Additionally, T cell responses have also been described. So, given the safety and the possibility of a response, we generally are still recommending the annual flu vaccine. There was a good number of studies on COVID-19 vaccines over the past 4 years at patients with antibody deficiency. And the good news is that these have been robust enough vaccines that even in patients with suboptimal or deficient responses to other vaccines, we're still seeing partial, if not decent responses to COVID-19's booster vaccines and boosters in patients with antibody deficiency. It's a little small here, but you can see that so on the right is receptor binding domain antibodies. That's the key kind of neutralizing segment within the SARS-CoV-2 spike region. And though patients with X-linked agammaglobulinemia generally didn't make a response because they have no B cells, patients with CVID in blue generally there was a wide spectrum compared to the normals in the patients who are otherwise healthy in green. But most patients made at least a partial response, and patients with specific IgA deficiency generally made a normal response. And then on the right is SARS-CoV-2 specific T cell response, and you see that these are actually even in patients with XLA are actually pretty robust. So again, it's another pretty good argument to go ahead and get the vaccine, and that even if you're not making great antibody responses, the other arm of your immune system should still be stimulated to generate memory. So current COVID-19 vaccine recommendations for our immunocompromised patients, and this is particularly pertaining to patients who are getting who have kind of moderate disease, is that they go ahead and is to get 3 doses if you've never been vaccinated before, age 6 months or greater. or if you have not received the latest updated 2014 vaccine, that you get at least one a minimum of 8 weeks after your last dose. So this covers the newest KP2 strain and KP2 and the newest KP3 now represent the majority of infections in the US, and they're fairly closely related. So the hope is that the new vaccine should provide cross-reactivity to it. There's also, besides mRNA vaccines, there's also now a protein vaccine made by Novavax as approved at age 12 or up. So that's approved for 2 doses if you've never received the vaccination, or 1 dose as an update would be adequate. Another important vaccination to consider in patients who are immunocompromised is the new Zostavax vaccine. So, so excuse me, Shingrix. Excuse me. Shingrix is a recombinant subunit vaccine. Zostavax actually is live. This is important because Zoster, although it can be troublesome in anyone. In immunocompromised patients, it can be more widespread. It can cause neurologic complications, or in rare cases, it can even cause disseminated disease. So it can be quite serious. And Shingrix is a recombinant glycoprotein E vaccine, so there's no live virus in it at all. It's approved for all adults age 50 or above, but it's actually now approved also in patients 18 or above who are immunocompromised. And it's given as 2 doses separated by a minimum of 1 month. Um, and all right, and moving on to so the RSV vaccines now we are now approved. So respiratory syncytial virus is a leading cause of bronchiolitis in infants as well as viral. Um, and we now luckily have three active vaccines as well as a passive vaccination, which Dr. Ward will probably talk about in a bit. And here we can probably, I mean, you can go forward one. There we go. So there's three vaccines approved. 2 of them are protein. Emrisia is an mRNA vaccine. All of them are approved basically in older adults, 60 or older. CDC generally is recommending for otherwise healthy older adults that it be considered particularly in the 70s. But Abrisa also is actually approved for pregnant women and recommended in the 3rd trimester to protect both her and also secondarily young infants. All of them are approved basically as a one-time dose, so they're not annual. They're not annual vaccines at the current time. But all of them in the clinical trials reduced severe RSV by up to 80%. So moving on then to mild to moderate B cell deficiencies, such as selective IgA deficiency or subclass deficiency. And these individuals, inactivated vaccines are safe and likely beneficial. And in some cases, really giving additional boosters is one of our main strategies for keeping these individuals well. And live vaccines also. Sorry, I mean, you can click forward. Live vaccines are usually safe and recommended as well. Lastly, I'll mention phagocyte nutrient deficiencies is a type of innate immune deficiency, including chronic granulomatous disease or leukocyte adhesion deficiency. In these individuals, we would not give live bacterial vaccines because they can be potentially dangerous. So that includes oral polio, oral typhoid, or BCG. Luckily, oral typhoid is really the only one that one would potentially be in contact with in the US, mainly in a travel clinic, but otherwise it's not. None of these are the standard on the standard vaccine schedule in children. But otherwise, inactivated vaccines and live viral vaccines are safe and recommended in patients with these disorders. Another important topic is vaccination after bone marrow transplant or gene therapy in patients with more severe types of primary immune deficiency. So current guidelines usually suggest resuming vaccinations anywhere from 3 to 6 months post bone marrow transplant, depending on the details of the transplant and the recovery. There's no real consensus presently from the transplantation consortiums and societies, but Generally, most programs go by patient's recovery, and particularly their ability to stop immunoglobulin replacement, as well as the recovery of their lymphocytes, mainly their CD4 T cells and B cells. Um, with regard to the COVID-19 vaccine, we generally would give that as early as 3 months after transplantation. Um, but inactivated vaccines are usually given first in order to determine if patients make a good response before moving on to more general catch-up. So another important factor is use of vaccines in patients who are on immunoglobulin and trying to determine whether or not it's safe to discontinue. Again, this obviously depends on the specific condition. Mostly we see this in patients with hypogammaglobulinemia who have not to date had a specific gene cause or have not met criteria for CVID, for instance. And these individuals' use of a vaccine that's not generally present in the general population in the plasma donors is useful. And it turns out the inactivated typhoid vaccine, which is type 5I, actually meets this requirement nicely. It's a one-time inactivated vaccine, generally very nicely tolerated. And Jack Muniz and team in Wisconsin showed that by testing antibody responses in patients who were still on immunoglobulin before and after typhoid vaccine, we could successfully discern people who had kind of clear known antibody deficiency versus those who had hypogammaglobulinemia without a known cause, and thereby so this is a way to determine if someone's B cells likely are still functioning even while on immunoglobulin and kind of add additional data to support stopping immunoglobulin and watching more safely potentially. Another common question that comes up is vaccinations for other family members when one or more individual has primary immunodeficiency. This often comes up, especially when patients are undergoing bone marrow transplant, for instance. Vaccinations in family are a really important way to secondarily protect an immunocompromised individual, and that includes live vaccines. They're generally safe for close contacts, including measles, mumps, rubella, and varivax. The transmission of these are very unlikely. Really, only notable is after the chickenpox vaccine in young kids, it's possible potentially to get viral lesions at the site of injection. So generally covering that with a dressing is a good idea, and avoiding contact with the area. Obviously, if you have a more widespread rash, then usually that individual should be separated from someone with immune deficiency. Generally, oral poliovirus can be spread, but that's not used in the U.S. I've noted it is used in the Middle East and is actually, unfortunately, one of the causes for the a poliovirus reversion that's being detected in Gaza, and one of the reasons that vaccination in that area has been so critical. Oral typhoid vaccine also is potential for spread, but again, unlikely that that it would be suggested except in global travelers. So with that, I'll mention so the CDC and AAP have great information about the vaccine schedules and special populations, as does the IDF. So that happy to happy to take questions after Dr. Ward's presentation.
All right, thank you so much, Dr. Keller. I'm gonna stop sharing, and Dr. Ward, I'll let you go ahead and take over with your part of the talk.
All right. So I'd also like to thank the IDF for the invitation to talk with everyone tonight here. I think this is a very important topic, so we're definitely happy to talk about with everybody. So again, we're going to be talking about arming yourself against these infections. I'll start off by mentioning that I participated in several clinical trials over the past few years, but none of these have had anything really to do with prevention or treatment of any of the infectious diseases that we're talking about here today. Really, we're going to be talking about how to help kind of prevent these infections. And the most important step, Mike has just finished talking about. So vaccination. It's a really big one. So thanks, Dr. Keller, for going over that thoroughly for the audience here. So we're going to be going over some other ways to help kind of avoid these infections kind of on a personal level and a kind of a micro level. First step is doing our best really to avoid exposure. If you can avoid it, then you're not going to get sick with it. And this really starts with some good kind of basic practices like hand washing. It's a very basic thing that we think of, but it's really important for controlling the spread of these infections. And it's also important to remember to clean commonly used surfaces. So things like doorknobs, the railings on stairs and things like that, they can be really filled with germs and viruses and all kinds of things. So make sure that these are cleaned very well, and that can do a lot to help prevent the spread of these infections. Okay. Improving the air quality around you can be very good as well too. So opening up doors and windows, let some of that fresh air blow in and can help to encourage the particles, the droplets that are in the air that are potentially infectious to get out. Also, exposure to sunlight and the chemicals in fresh air can actually help to inactivate those and make the air less infectious potentially. One thing you can do inside your house or your office or whatever is set the HVAC unit, instead of having it on auto, turn the fan so that it's always on. So that makes sure that the air is constantly being circulated in the indoor space. And that means it's sending that air through the filters, which then can hopefully take some of those droplets and infectious particles out of the air column. There are lots of personal HEPA filters that you can take around with you, set up at home or in your office, etc., that can help to clear the air in a kind of a localized area as well. And then, of course, if possible, if you're going to a large gathering, trying to move those parties outdoors can be helpful. Again, getting that fresh air going through. Now, admittedly, we're coming up into kind of the fall and winter months here. So depending upon where in the country you live, this may be a little bit easier or not. But whenever possible, try and remember to get the fresh air from outside. Okay, masks can still be very important. I think it's really one silver lining of the pandemic was I think that the kind of the stigma around wearing masks here in the US has decreased. And I think it's a little bit more considered more of a regular thing now. So masks are very important. It's important to choose the best type of mask. So certain masks are more effective than others. So cloth masks can be helpful, but surgical masks will be better. And KN95 masks will be better than that. And N95 masks will be even better. So choose whatever mask is available. It's the best type of mask that's available to you. Once you get that good mask, make sure you learn how to use it. So it's real important to make sure that the mask is covering your nose and mouth completely. There's certain ways to put on the K95 and N95 masks that make it a lot easier, and so you don't potentially spread stuff from the outside of the mask onto your hands or face. And then it's always important to check for air gaps. So give a couple of good breaths out, blow out, and hold your hands around the mouth, around the mask to see if you can feel any air coming out, because those are areas where contaminated air potentially could get in a little bit more easy. Physical distancing is still a good practice to practice. It seems kind of obvious, but you want to avoid people with active symptoms. So if someone is hacking and coughing, you may want to kind of steer clear a little bit if you can. Avoid large crowds because you're more likely to be exposed in close proximity to someone who might be infectious in large crowds. But remember, there's no single one safe distance that you can always practice. The distance at which some of these droplets and things will carry in the air is dependent on a large number of factors, like the amount of sunlight, the temperature and humidity, the direction and speed of the wind, etc. So just try and use your best judgment, but again, there's no one size fits all for this. And then finally, testing. Testing can be very, very helpful. I think it's a good idea to have test kits available in advance. So go get them when you're not sick, so you have them at your home or whatever, and you can test before you really need it. The other thing about test kits is they can be helpful for gatherings. If you know you're going to have a family gathering during the holidays, for example, if you can convince your family to test and make sure everyone's testing negative, that can go a long way to ensuring that everyone stays healthy during those gatherings. Okay, so a little quick word about hand washing. I know this seems pretty simple. I think most people kind of, intuitively know how to wash their hands, but there's a couple of points here. So one, make sure you lather up your hands and really scrub them for a good 20 seconds. That's key. The more exposure and more time the soap has on the hands, the more germs and viruses and bacteria are going to be removed. So try and go for 20 seconds. A good rule of thumb is to sing the ABC song in your head or sing the happy birthday song twice. Both of those will end up being around 20 seconds. And of course, then rinse and dry your hands after that. A hand sanitizer is a pretty reasonable second alternative if you can't wash your hands. Just remember that you need to use a hand sanitizer that's at least 60% alcohol and you need to wet the entire hands. So if you use just a tiny little drop and it evaporates really before you even get a chance to rub it in very much, that's not going to be very helpful. You need enough to wet the both hands so that the alcohol has a chance to kill the viruses and bacteria. Also keep in mind that it may not be as effective as hand washing, particularly if there's visible soiling on your hands. If you've got snot or goop or dirt or whatever, hand washing is still going to be the better way to go. All right, so now we're going to talk a little bit about passive immunity. And this means that you're really relying on immunity that you get from others. So not in the herd immunity sense, but actually directly getting it from somebody else. Okay, so for those who have immune deficiencies in which immunoglobulin replacement is indicated, making sure that you get your IVIG or sub-Q IG can be very helpful. So immunoglobulin does contain what's called broadly neutralizing antibodies to different viruses. And this can, in some cases, confer protection, at least some protection, against new strains that haven't come up yet. So this was a study done by a group looking at a model of influenza in ferrets, actually. And so they gave the ferrets a dose of IVIG from a year prior to the dose of the flu strain from that year. And those that got placebo, as you can see, had worse activity scores, which is kind of analogous to a symptoms score. So the darker the color, the worse the symptoms were. So those that got IVIG, however, even though it was from a year prior, still had a significant amount of protection. Okay. Now, it's important to note, though, that these broadly neutralizing antibodies, the amount of them that we do see in IVIG is generally low. And while this example I showed here illustrates that this beneficial effect can be present, it's not going to be present in all preparations. It's not going to be the same for all strains of the virus. So it's definitely not a substitution for vaccination. So still go out and get those flu vaccines like Dr. Keller had just mentioned. Okay, but if you have an immune deficiency where immunoglobulin is indicated, make sure that you get it on schedule and keep it up because that will be very, very helpful. Okay. Now, beyond that, there have been some products that have been developed to help give passive immunity to COVID. So earlier in the pandemic, there were a number of products that were monoclonal antibodies or convalescent serum that were developed to kind of help the spread of COVID and help protect people. Unfortunately, those products, the virus ended up changing and mutating, and those are no longer as effective against the newer strains. But we do have a new product called pemivibart that was approved several months ago, in April, I believe, for the prevention of COVID-19. So it's indicated or approved for people that are 12 years of age or older. It's an infusion, so kind of like IVIG, it's an infusion. You'd get it once every 3 months, and in order to qualify it, you have to have a moderate to severe immunocompromising condition and be unlikely to respond to vaccinations. So that second point is important because some severe, potentially severe immune deficiencies like chronic granulomatous disease, the antibody production is relatively preserved, and those patients wouldn't necessarily benefit, or this wouldn't be indicated for them. It's also important to note that this is not for treatment or for trying to keep you from getting sick after you're exposed. This is really kind of a preventative treatment. Now, pemivibart is available in a number of different centers. It's mostly available, I will say, kind of in the middle of the country, so kind of the Midwest and kind of maybe the Southwest kind of portions of the country. I just checked the other day, the website for the company that makes it, they have a tool that will allow you to check if there's a center near you. There's not many on either coast, unfortunately. But if there is a center near you, then it could be a good beneficial tool to help prevent COVID. Now, there is one caveat to that. So pemivibart targets a region of the COVID virus, the spike protein, that is very, very well conserved. That being said, there is some preliminary evidence, and by that I mean it's not yet been peer-reviewed, suggesting that the ability of pemivibart to neutralize the newest strain, the KP 3.1.1, is reduced by a significant amount. And you'll see here, this is data from the CDC that unfortunately that particular strain is becoming quickly kind of the predominant strain. Now, I don't want to say that there's no use for pemivibart, because also the study that looked at the neutralization used a type of screening test that is known to kind of maybe underestimate or potentially underestimate the ability for the antibody to neutralize the virus. But it's something to keep in mind. I think it certainly would be potentially more helpful to get those infusions now coming in. And as time goes forward, we'll see whether or not it retains its helpful ability to prevent the KP 3.1.1. Okay. So for RSV, so Dr. Keller highlighted this a little bit. We do now have a passive immune preparation for RSV. It's a medicine called nirsevimab. It's a monoclonal antibody that targets the RSV virus. It's indicated for all babies who are less than 8 months old if their mother didn't get the RSV vaccine before they were born. It's also indicated for children less than 19 months old, so particularly that second year, that second RSV season, if they have an increased risk, such as those babies that have inborn errors of immunity. Okay. It's given as a single injection, and it's given sometime during the RSV season from October to around March. Okay. There's also an antibody called palivizumab that has been around for a number of years. Now that the new nirsevimab is out, actually, nirsevimab has a much higher ability to neutralize the virus. And so really, we only recommend palivizumab if someone is unable to get the nirsevimab. There's a few exceptions. For example, palivizumab is given as monthly injections through October through March. And so if someone gets injections October, November, December, January, February, there's not really necessarily a need to get the nirsevimab injection, that last injection in March. You've already had those 5 palivizumabs, just stay with that. But otherwise, generally speaking, if nirsevimab is available, that's what we're recommending to be given in most cases. All right. So next, I want to talk a little bit about what we call post-exposure prophylaxis. So this is how to protect yourself after you've known to be exposed to your coworker that was coughing during the meeting, or the kid sitting next to you at school who is sneezing all over the place. So with influenza, we actually have a number of very, very good options to help prevent infections after known exposures. So oseltamivir, zanamivir, and baloxavir marboxil have all been shown to really pretty effectively reduce the risk for developing symptoms after exposure. There are a few little differences. You'll note here, oseltamivir is approved down to age 3 months, so it can be used even in young kids and infants, whereas the other two are mainly for older children and adults. Okay. Zanamivir is inhaled. It's a little inhaled medicine that you breathe in. And because the medicine is going into the lungs, it's not indicated for people with any pre-existing lung conditions like asthma, COPD, or other kind of issues where the inhaled medicine might trigger exacerbations of those issues. The other thing to note is that baloxavir marboxil is just a single dose, whereas the other two have to be taken consecutively for a total of 7 days. Okay. Now, all of these, you have to start them within 48 hours of exposure. So you don't want to kind of sit on your heels and just kind of wait around and see what happens. If you know you're exposed, you should go and get started on these medicines right away. Okay. And one other kind of tidbit to mention is that if there's someone that's exposed in an institutional setting, like in a hospital or in a long-term skilled nursing facility, for example, instead of the 7-day duration, we recommend that be continued for at least 14 days, plus at least 7 days after the last known exposure. Okay. Now, even though we have some good options for treating COVID, we've not really had good success in preventing the development of symptoms after known exposures. So there's a recent trial that looked at one of the good medicines for COVID, which I'll speak about here in just a second. They tried to use this as a post-exposure prophylaxis. So after people were exposed, they gave them either a 5-day or 10-day regimen. And unfortunately, there was really no statistical difference between any of these groups. So it looks like it doesn't really help to prevent the development of symptoms after exposure. Okay, so good options for flu, not so good for COVID, and unfortunately, we don't really have any options for RSV. Okay. All right. So what do you do if you get sick? This is the next part. Again, we've got some good options here for some of these disorders. For COVID, one of the things that happened during COVID is there was a large mobilization of research to kind of help develop these antiviral medications, and there's several of them that can be very effective in controlling COVID. So there's nirmatrelvir, which is boosted by a known booster called ritonavir, remdesivir, and then molnupiravir. Okay, those are a mouthful, but nirmatrelvir is indicated for anyone 12 years of age or older. It must be used within 5 days of exposure. And it works pretty good. The one kind of caveat is that it does have a lot of drug-drug interactions. So depending upon what other medicines you might be on, there might have to be some modification of the dose, or sometimes your provider might ask you to pause certain medicines if they're not essential, for example. But definitely talk with your providers about your other drugs that you're taking if you're going to end up starting the I can't say it, Paxlovid, excuse me. Now, remdesivir is a very good medicine, and it is been used a lot in inpatient settings because it's an intravenous medicine, but it can be given as an outpatient as well too. really the dose and duration of this kind of depends upon the severity of the disorder. So for mild or moderate cases, we might only use it for a total of 3 days. However, if someone is hospitalized in severe condition, the duration may be continued for 10 days or longer, depending upon how the patient is doing. And then finally, there's molnupiravir. It's approved only for adults, and it is similar to nirmatrelvir in that it is an oral preparation used for 5 days. It doesn't seem to block the replication of the virus quite as well. So typically, we only use it if there's some contraindications to taking the nirmatrelvir. So it's kind of a backup if there's not another good option for you. Okay. Now, one question that always comes up is the question of rebound. So rebound symptoms are very possible with any of these medicines. What rebound is you start to get symptoms of the disease, COVID. They may improve on the medication, but once the medication is stopped, then the symptoms may come back again. And the fortunate thing is, so far, the research that's been done indicates that, the risk for developing severe COVID symptoms, so respiratory distress or other things, if you get rebound, is really, really non-existent. So it's very, very low. And so if you do get some rebound symptoms, they're typically very, very mild and much better than they would have been had you not taken the medicine in the beginning. All right, for treating influenza, again, we have some very, very good medicines. Several I've already mentioned, the oseltamivir, zanamivir, and baloxavir marboxil. There's one other one I'll mention called peramivir. So all of these need to be started within 48 hours of developing symptoms, ideally. So you need to really kind of start them again soon as you start to get any symptoms of influenza. Oseltamivir is indicated for anyone of any age, even down to infants, and even in some cases neonates or prenatal premature infants. So zanamivir is only indicated for treating patients 7 years of age or older. Baloxavir for 5 years, and peramivir down to 6 months. So peramivir is an intravenous, so it's different from all of the rest ones, but it is given as just a single dose. So it's a single treatment, and then it can help to improve the symptoms thereafter. Okay. For all of these, the viral resistance to these medicines has remained pretty low over the last several years. So it's a good sign. And all of these should still be very, very efficacious with all of the flu strains that have been circulating already and are predicted to be coming in the future. So there are some older medicines. There's a medicine called amantadine and one called rimantadine. These were helpful years ago, but really not so helpful anymore, as really nearly all of the influenza A strains that are encountered nowadays are resistant to these. So it's really not even worth trying them in most situations. So for treating RSV, at this point, we're kind of all going off-label. So none of these are FDA indicated. So treat this all as kind of just information and off-label use for medicines. Ribavirin is a medicine that can be very helpful. It's broadly active against a number of different viruses, including RSV. We typically reserve this for patients that are hospitalized, that have very severe immune deficiency and or very critical illness due to the RSV. The reason is that this particular medicine has got a lot of toxicities that kind of limit its use, not just for the patient, but more so even for the caregivers. So a lot of careful handling. The nurses have to treat it almost like a chemotherapy agent in some cases. So it can be helpful, especially in severe critical illness, but beyond that, it's not kind of worth the risk in most cases. I'll also mention that palivizumab has been used in treating RSV in a number of case reports. So there are more than a handful of case reports that suggest that it could be helpful in severe RSV cases, particularly in people that are immunocompromised. That being said, there was a trial that was done a couple of years ago looking at its use in the general population. And as a treatment in the general population, it didn't really seem to have much benefit. So again, this is something that your provider may talk with you with in special situations. It may or may not be helpful, but it's something that has been reported before and can be in the conversation. Okay, so just to sum up here, prevention is going to be the most important. So definitely go get those vaccines, the flu vaccine, the COVID vaccine, and the RSV vaccine, if they're indicated for you, is going to be very, very helpful. Avoiding exposure whenever possible is also a key point here. So try and steer clear of those germs as much as you can. And for those in which it's indicated, pre-exposure prophylaxis may be quite helpful. And so you can look to seek those out. Okay. For influenza, we've got some good post-exposure treatments. So make sure that you talk with your healthcare providers if you start to get symptoms after a known exposure. Even if you don't get any symptoms, ask for the treatment to help prevent them. Okay. And then, of course, for COVID and influenza, we definitely have some great medicines that can help treat the active infection and reduce the severity of symptoms. And even for RSV, although that's definitely a more specialized situation that requires discussion with your healthcare providers. Okay. So the CDC has some fantastic resources for all of this. There's some very general guidance about prevention of respiratory viruses of all kinds, as well as specific guidance about treatments and available prevention measures for COVID, flu, and for RSV. And with that, thank you guys for tuning in, and happy to answer any questions.
All right. Thank you so much, Dr. Ward. I'm going to go ahead and pull mine back up. All right. So I know you and Dr. Keller have been in the Q&A box kindly answering some of our questions, but some of them I am going to consolidate and re-ask just because they were good questions, and I think the entire group would benefit from hearing your responses. So forgive me if there's a little bit of repetition, but with that, we are going to go ahead and get started with the Q&A. All right. So again, this is one, Dr. Keller, I think you covered this, but we're going to ask it to the group so everyone can hear the answer. This individual wants to know, it is my understanding that there is no lab test that shows the protective levels of the COVID-19 vaccines. Is that true? And if so, is there any work being done to change this?
It's a great question. I can press start. So when we talk about levels of antibodies, there's a couple of important things. Many tests really will look at total amount of antibody. Really depends on the type of test that's being done. During the COVID-19 pandemic, the medical community realized that that antibodies for the receptor binding domain on the spike protein were the antibodies that were most important in neutralizing the virus. So when I say neutralizing, I mean basically that the antibody, the protein, is binding the virus in such a way that it's no longer infectious. That's really the gold standard for an antibody that's really effective against a given, a free virus. So we actually can measure the amount of that receptor binding antibody. Not all the commercial labs necessarily are doing that, but the absolute cutoff for protection really depends. I mean, it differs by individual test. Generally speaking, though, I mean a response that basically is within the reference ranges for a given test usually would reflect that you have a good immune response to it, and likely you have some correlative protection. So that said, I mean, we generally are not, we wouldn't necessarily kind of alter what we're doing. Like for instance, we would not recommend more vaccines to a given individual based on a given test. Since again, these vaccines also generate a T cell response that's a little harder to track.
Thank you. All right, next question. Again, one that I think everyone would benefit from hearing the answer. Should vaccines be taken relative to IVIG infusions? And should this be done at the midpoint, in between infusions, or does it not matter?
Um, yeah, so there has been some concern raised about getting live vaccines in relation to IVIG, in which case the live virus is needed to kind of spread to generate the immune response. And so for those, there's some kind of indications to not give it. Um, for other vaccines, it's not quite as clear that it makes as much difference. And there people get a good response to a number of inactivated vaccines, kind of regardless of being on IVIG or kind of when. So I would say that generally for live vaccines, there's a potential issue there, but for the vaccines, it shouldn't be as important.
Thanks, Dr. Ward. All right, next question. So, if I'm a patient and I receive protection from IG therapy, why are vaccines necessary? I'm still confused and in need of help. Similarly, if one does not mount a response to vaccines, is there any point in getting them?
Yeah, this is a great question. Really, the only and this individual's right, and generally, so when we give immunoglobulin products, we're giving pooled plasma from thousands of donors. Catch is those donors, and generally for that product that you're looking at in front of you, be it sub-Q or IV, is from plasma that's generally been drawn and processed anywhere from 9 to 10 months, or even perhaps even longer, previously. So it's never totally kind of up to date, immune response from a person who is seeing the same circulating viruses that are happening in that season. So that's one reason that we still usually would recommend that the uh, annual vaccines for influenza and for COVID, because again, these are rapidly changing viruses where the government health officials and the vaccine makers are really actually having to do some investigation work in terms of what's circulating, for instance, in Asia and in Australia, in the and during the earlier in the year before our influenza season starts. So it's a bit of a guess, but nonetheless, it's more reflective of the current viral mutations and the current shift that's happened in the virus. And obviously, it's the efficacy varies year to year, but even though there is, for instance, antibody against COVID-19 in immunoglobulin, having that additional booster, as best we can tell, is safe in most people with different types of immunodeficiency. And between the fact that it's that these vaccines usually do engender at least partial response in many individuals, and also can induce a T cell response that you wouldn't get from immunoglobulin, it still may benefit you and is very unlikely to do any harm.
Thanks, Dr. Keller. All right, next question. This individual says, before I was diagnosed with PI, I used to get live vaccines as a child. Broadly speaking, should I be worried about that? For me, that was over 50 years ago.
So the answer is in most people it doesn't seem to cause any long-term sequelae if the vaccine was originally received and did well with it. So many people that develop common variable immune deficiency, for example, they develop it later on as an older child, teenager, or adult, and many of them seem to do pretty well. That being said, like Mike highlighted, there are some vaccine strains that can cause some problems. Usually this is in more severe immune disorders that may present at an earlier age. But for most people that present kind of later in life with an immune disorder, generally speaking, the risk is pretty low.
Luckily, I'm not aware of any bad rubella-associated granulomatous disease in CVID patients, at least not to date.
Thank you both. All right, next question. Is there a relationship between COVID-19 vaccines and long COVID? Could vaccination help prevent long-term effects?
Yeah, great question. Sorry, go ahead.
I was just going to say, yeah. So there has been some suggestion that it does seem to help prevent the development of long COVID. So I think, we're still learning a lot about COVID, and we don't know entirely what causes long COVID or really what long COVID is. But there are some hints that getting the vaccine does seem to decrease the risk for developing long COVID. And so, yeah, so in that regard, it may be helpful.
Yeah. And there's a lot of work that's being done on what's being termed kind of infection-associated chronic illnesses, like long COVID and chronic fatigue syndrome and related diseases. And best we can tell, I mean, part of it seems to be some mimicry between a given infection and self proteins, but certainly doing anything you can if you have long COVID to prevent a reinfection is a really good idea.
Thank you both. All right, next question. This person wrote, I'm a person who has had many, in all caps, COVID vaccines and boosters. Should there be a period of time that I should be waiting between vaccinations?
So yeah, we certainly sympathize as healthcare providers, and obviously, in many people that day after can be a very ibuprofen-driven day. But that said, I mean, if you tolerate vaccines, if you've tolerated prior vaccines well, and as long as 8 weeks have elapsed since your last COVID-19 booster, having the KP2 updated vaccine is probably a good idea because it should provide more accurate protection against the new circulating strains. So the short version is, if it's tolerated well, it's probably a good idea to get the newest run at a minimum of 2 months after your last vaccination, or for that matter, if at least 2 months after last infection, if you had it.
And I'll add too that there's some data to suggest that the severity of kind of the secondary consequences that you feel from prior vaccines may not be the same with each subsequent vaccine. So some people might have a very bad reaction after their first COVID shot, for example, but do great after their second, or vice versa, may sail through the first one and feel like they kind of were run over for their second one. So it doesn't necessarily mean that you shouldn't get that booster later on as well too.
Thank you.
The fatigue and whatnot is what we call reactogenicity, and it's not allergic as much as it is actually immune activation. And if any, in many cases, actually, that kind of reflects that your immune system is doing what it's supposed to do.
Thank you. All right. What is the latest information on monoclonal antibody treatment related to COVID-19? Is that still being advised for immunocompromised patients? And if so, how could one locate or access
Already
this
covered
type
this
of
now.
treatment?
Yes. So yes. So pemivibart is the monoclonal antibody that's currently under authorization for the emergency authorization for prevention of COVID-19. So it is available. Like I mentioned, it is not available everywhere for reasons that kind of are not entirely clear to me, but it mostly seems to be available kind of clustered in the center of the country. But it is still available, and it does seem to still be active against most of the currently circulating viruses, with the caveat that the newest KP 3.1.1 may be a little bit less effective. But while there's still other viruses that it can effectively neutralize, I think it's still a good option to consider.
Thank you both, and apologies, I had to turn my camera off. I wanted to make sure we weren't going to lose connection here. All right, next question. What would be an effective antibody level after receiving the COVID vaccine? I know there is probably not a normal here, but trying to get some sort of idea as to what that might be.
Yeah, that's a tough question. I think it really depends a great deal on the specific test that's being done. Like children's, we use the diazorin receptor binding IgG. Um, it really depends. So each lab usually establishes their own normative values for a given population. So I would talk to your provider and look at the results relative to that specific test that's being done to kind of get a better sense. But I'd stress also though that it's only half the picture because again we can't routinely test T cell responses to the mRNA vaccines, but they're definitely present. And so as that latest European review that the figure that which I put in the in my slides showed T cell responses actually are more uniform than antibody responses in our PI population.
Thank you so much. All right, next question. Do you recommend that a senior individual receives the double flu? This individual has CVID, and or other primary immune deficiency. This person shares that they found that without the double flu vaccine, they get the flu, but it is becoming harder and harder for them to get authorization to get a double flu vaccine at the pharmacy, and they have to go to their primary care provider. Is this advised,
Yeah.
I guess is the question.
Yeah. So in general, the evidence was pretty good to suggest that older individuals, the high strength, quote unquote, flu shot did seem to elicit a little bit better protective response. Um, and so, yeah, in general, I would say we do recommend that if you're in the age group for which it's indicated, that it is good to get. There's been some talk about whether people that are younger than that, but who are immunocompromised, should get the high strength. And to date, there's not really a lot of data to support it one way or the other. And it can unfortunately be a little bit tricky to get those covered if you're not in the age group. But if you are in the age group, I do think it's a good idea to try and get it if you can, because it does seem to help kind of boost the protective levels a little bit more.
Thank you. All right, next question. This one's very interesting. Do you know if infants can shed the virus or a virus from live vaccines through stool? For example, if a PI patient has a child who is still in diapers, and this child received live vaccines, would diaper changes be a concern for the PI parent for a time period after the child has recently been vaccinated?
Great question. That is the case for Rototec for the rotavirus vaccine. Um, that said, it's inactivated. So, I mean, in most individuals with PI, it really shouldn't, it really only be a risk to people with fairly severe disease. So it wouldn't give me pause, for instance, in a patient with antibody deficiency to give it to their children. Um, and definitely, rotavirus can be quite, even in well children, rotavirus can be quite nasty in infancy. It's responsible for over 100,000 hospitalizations and ER visits annually in the U.S. So for my 2 cents, I think you could talk to your provider certainly and determine if they think if you're too immunocompromised, you can discuss with your provider and your pediatrician if it's worth holding off. But in most cases, it should be safe. I don't know if that's a word. If you
Yeah, no, I agree. And I was going to add the other one that might be of concern would be the oral polio virus or a vaccine that we don't routinely give here in the US anymore. But yeah, so I think that's less of a concern for our population here in the US, but that one potentially could be spread too.
Thank you both. All right, next question. Is there any science or legitimacy surrounding the concept of microdosing exposures? This person shares with masking, I seem to get sick more in the summer, and this is a person who has PI.
Hmm. So, it's a good question, and I have to say I'm not aware of a lot of good evidence about microdosing. I think there was a um, interesting kind of a side note about one of the Olympian athletes who tried to microdose a little bit before swimming in the Seine during the Olympics. Didn't seem to work as well for him, but so yeah, but I'm not entirely sure if there's a lot of good evidence to support it. But I think in general, um, I think, trying to prevent some illnesses that could be associated with severe disease, I think is going to be the best bet. And I would say to steer clear of it. So I would generally would say steer clear of COVID if you can, and things like that. But I don't know, Dr. Keller, if you have any other thoughts.
Yeah, for a lot of these viruses, it's just, I mean, if you either had enough exposure that you get sick or you didn't, and you're not, generally speaking, it's unlikely that you would generate an immune response if you didn't actually get an infectious dose. So I would agree. I think I mean, it's the case that when you stop masking for a period thereafter, you are apt to get more sick than you were. I mean, that was pretty much the entire story of 2022 for most of us, um, even in otherwise healthy individuals. But otherwise, I think my only suggestion would be just talk to your provider about making sure that you're um, optimizing all other preventative medicines, be it immunoglobulin or preventative boosters or what have you.
Thank you both. All right, we have a couple more. This individual wanted to confirm what the vaccine schedule is for RSV. So they wrote that they thought they were supposed to give it every 2 years, the RSV vaccine. Is that accurate, or is it a one and done situation?
Depends on the type of vaccine we're talking about. So the active vaccinations, um, so those are basically a one time. Um, so to the date at least. Again, they're all three of them are new. So and and for that matter, I mean, importantly, there's fairly limited information on the active RSV vaccines, duration of efficacy in immunocompromised patients. Um, but that said, I think for older individuals, if you're eligible for it, they certainly are safe and seem to be effective in reducing the chance of a serious case. Um, for the passive dosing, I'll probably tag team out for Dr. Ward for that one.
Yeah, yeah. So for the passive dosing, the newest the Beyfortus, the brand name is recommended to be given once for most infants, and then for those infants that have primary immunodeficiency or other reason to have severe disease, they can get a second dose as long as they're younger than the 19 months. So it's just kind of one and done or two and done potentially for that. And the older monoclonal antibody, Synagis, was given once a month. It was an injection once a month during the season. So that's a little bit of a different protocol. But again, that's not one that we recommend doing right now if the newer Beyfortus is available. And right now, they're so new. There's almost zero data on whether or not the passive immunity preparations like Beyfortus will be helpful in older people, older children, or kind of middle-aged adults, or those that have PI. We just don't know yet. In theory, you could say that it probably should be helpful, but again, there's zero data. Unfortunately, it's going to be a very, very tough kind of hill to climb to try and get that approved by insurance to pay for it or dispensed by a pharmacy if you're not in that first year or second year of life.
Thank you so much. And this is going to be our final question of the evening. Can you tell us what contributes to repeat COVID infections, and is there any literature to explain why these occur?
Sorry, Emma, we lost you for a moment there.
Oh, can you guys hear me okay?
Yes, we can now.
This person wanted to know what contributes or what is the cause of repeat COVID infections, and is there any science literature that's been achieved to look at this topic?
Sure. At a very kind of overview, just kind of a basic level. Um, Repeat infections are a combination of kind of changes in the virus and duration of immunity. And so, as we had, the various Greek letters that's COVID has gone through, and to achieve to get to now our current Omicron flirt variants. There's been specific changes in the virus that have allowed it to evade both different types of antibody responses and to some degree even T cell responses. So at the same time, our immune system is kind of, again, obviously we're getting exposed just through either vaccination or when we occasionally get infected. So we're getting kind of snapshots of that virus as it's changing over time. Um, and so the plus side is a lot of responses that we make generally at least give you some protection against the newer variants, but they may not. So it's largely why at least our hospitalization rates have fallen pretty dramatically, but they may not. There's very big difference between the level of immunity in order to keep you from getting critically ill versus the level of immunity to keep you from getting infected at all. So that's why the duration of protection against infections is relatively short after vaccination, generally about 3 to 4 months, sometimes maybe a little longer in some individuals, whereas the protection against more severe disease is more lasting.
Yeah, so I'll add that it's very similar to why we can get flu. So in a similar kind of aspect, the flu is changing, the COVID virus as well, and the duration of the immunity that we have to those viruses, like Dr. Keller was saying, is not forever, unfortunately. It'd be great if it was, but um, it's one of those things. It's constantly evolving, and so we're gonna be constantly kind of an arms race to try and figure out who has the virus better virulence, so to speak, ability to attack us, and then our immune system has the better ability to evade it, and it'll keep going kind of in that arms race kind of in perpetuity.
Wonderful. Well, thank you so much to both of you for joining us this evening. Thank you so much to our audience for your thoughtful questions and being so engaged during the Q&A. Before we close out, I do have a few more slides just going over some resources and upcoming events with IDF, but Dr. Keller and Dr. Ward, you've already dedicated your evening to us. If you have lives to get back to, we totally understand. But we want to make sure you know how much we appreciate you joining us this evening and having you present this talk for our first Decoding PI series. So thank you both so much. All right. All right, everybody, just a few more slides to get through before we close out. Be sure to check out our website for additional resources, upcoming events, and more. All IDF materials are free to access, print, or have mailed directly to you. And we're going to share the link to our website. That's primaryimmune.org. That's where you can find all of our resources, upcoming events, and more. If your question was not answered this evening, you can contact our resource navigator through Ask IDF. She's wonderful. She will personally connect with you to tackle your question and direct you to appropriate resources. And the link to access Ask IDF is also being shared in the chat. You can even take IDF on the road with our engaging podcast series. You can find programs like Bold Conversations and Undiagnosed by searching for the IDF podcast in your platform of preference. I'm also really excited to share about some of our newest resources. Very timely with this event is that we have updated our flu and COVID pages on the website, and we definitely recommend that you check them out after the webinar. The links to find those pages are listed on screen, and they're also being shared now in the chat. We also have a brand new immune system self-assessment tool. I'm really excited about this one. This is a 10-minute questionnaire you can take from your laptop, from your phone, and it can be used to help identify potential signs of PI and help guide a conversation with your medical provider. Again, the link to access the new survey is on screen and also being shared in the chat. And finally, we have the healthcare provider template for letters of medical necessity for IG therapy. This is a tool that can be used by your provider to advocate on your behalf if insurance is denying coverage of your IG replacement therapy. Again, to access that template, you can either download it directly at primaryimmune.org, and the link to access is also being shared in the chat. Are you looking for ways to connect with others who are navigating life with the PI? You might be interested in joining one of our many Get Connected groups. These groups are free, virtual, and volunteer-led opportunities to connect with others with PI all over the US. We offer location-based groups, so what city you live in, or nationwide groups. And because the groups meet virtually, you can join any group, time, leader, that works for you. It's totally up to you. And we also have a brand new Men with PI group if that's of interest to anyone. IDF's new documentary, Compromise: Life Without Immunity, offers an intimate look into the world of those living with primary immunodeficiencies. Screenings and private watch parties are available now by requesting the film at primaryimmune.org/compromised. And the Walk for PI continues this fall with several more in-person events and even a virtual option. And it looks like we have the St. Louis Walk this weekend. Visit the Walk for PI website, linked in the chat, to register and build your team. We offer webinars each month, and we hope to see you at one of our upcoming programs. Here's a look at some of our upcoming programs in the next few months. And on that note, if you have a great idea for a topic or a presenter that you would like to see at a future IDF event, we want to hear from you. Visit the link in the chat to submit your idea for consideration. And with that, before we close, we want to thank our incredible sponsors for supporting our education initiatives here at the Immune Deficiency Foundation. We appreciate everyone's participation this evening and want to give a huge thank you again to Dr. Keller and Dr. Ward for their time and expertise that went into these talks this evening. We hope to see you at our next webinar on October 24th. Take care and have a great evening.